Like me to introduce you?
Yeah, go ahead.
Okay. The next talk is Unexpected Features of the Dark Proteome of Structural Biology with Nelson Pardiago, Julian Heinrich, Christian Stolt, Kenneth Saber, Michael Buckley, Bruce Tabor, Beth Signal, Brian Gloss, Christopher Hemming, Burkhard Rost, Andrea Schefferhans, and Sean O'Donoghue. And Nelson Pardiago will give the talk.
Actually, Sean O'Donoghue will give the talk. Sorry.
Sean, I'm sorry.
I don't know
Sean,
why there was
you're
a confusion
highlighted.
about that.
Sean Donoghue will give the talk.
Okay. So how do we study what we don't know? And that's particularly, notice this, everything's fine. This is a blank slide. It's supposed to illustrate the things that we don't know, right? And this is a problem for me because many people may know that I advocate the importance of visualization, and we run this meeting that I'm just going to briefly mention that and I think that basically I believe that the key limit of discovery in the life sciences is we're doing visualization inadequately currently, and we need to up our game, and that's what this meeting's all about. And we're from Sydney. Most of us, in case you needed an excuse to come to the world's most beautiful city, we're running the 8th International ISCB conference next year in June, and it's coinciding with an event in Sydney that is really awesome, where there's all these artistic and informative things happening around the city. So consider to pencil that in. But the topic at hand, yeah, how do we approach the unknown? How do we visualize it? Well, one way is to first look at what we know. And so I'm going to explain a little bit the first part of the talk about Aquarium, a resource we've been working on for over 7 years. And that essentially tries to map all known protein structures and everything we know about protein structures. So we know that there's a lot of structures out there. In fact, we've just gone over 120,000 entries in the PDB, which is a landmark in terms of our molecular understanding of the processes of life. But this gives us a new problem. You know, it's 120,000 structures to look at. So, and even worse than that, those are the experimentally determined structures, but we know we can model a lot more. So what we set out to do was to actually systematically model kind of everything, or at least a subset of everything. And when we started this project early on, we looked at the CASP meetings. And these meetings rank the methods that predict structure. The particular category we were looking at was the automated detection of a template by homology modeling. And in the 2010 CASP, the clear winner was a method called HH Blitz. And so we started working with this tool. It's a fantastic tool. And midway through the project, the next CASP results came out, and it had slipped and was no longer the outright winner. And we were worried we might have to sort of redirect our project. But then we realized, looking more closely, that everything that was higher ranked used HH Blitz for its seed method and was at least nearly 400 times slower. And since what we had in mind was a very large scale modeling, it was clear that HH Blitz was for us the best trade-off of accuracy and performance. So what's the magic of HH Blitz? Fairly simple, sequence alignment. But the trick is, in addition to taking the sequence you don't know the structure of, finding all similar sequences, building an HMM, you take the sequence that you do know the structure of, find all similar sequences and build an HMM for that. And now you don't match a sequence to an HMM, you match an HMM to an HMM. And the magic of HH Blitz is it manages to do that very quickly. And the effect is you can take the 110,000 PDB structures that are out there, map them systematically onto all 500,000 Swiss prot sequences, for example, finding wherever they fit. And because you go down to very low levels of homology, we get up to 50 million template models. So in a sense, we've made the problem worse. We're dumping onto the community 50 million models now. We have spent quite a bit of time we're working out where we should set these thresholds and cutoffs. That's the receiver operator curve for those of you who need to see such a thing. But yeah, we're making the problem worse because now not only 120,000 experimental structures to look at, but over 50 million model structures to look at. And to make those accessible, we built a web-based service that makes these models easy to use, easy to find, called Aquarium. We published it last year. And just to sort of step you through, we've paid a lot of attention to making the data in Aquarium sort of center stage, easy to find, easy to use. And just to build you through how the core view works, Aquarium is actually not structure-based, it's sequence-based. And by a protein sequence, I mean an N to C terminus full-length wild-type sequence from Swiss prot. Here's one illustrated here. And It systematically finds all matching structures. Of course, this relationship between structure is not one-to-one, it's one-to-many. So there can be different structures matching different parts of the sequence. And let me show you how the Aquarium web resource looks like when you get there. So as mentioned, it's really about sequences, not about structures. Essentially, we use structures as a way of annotating sequences. Now, each sequence, the number of matching structures can be quite large. For this particular case, we have 145 PDB structures that match down to a very low level of homology to any part of that sequence. And we create what's called a concise visual summary of those 145. To make it concise, we cluster them. So for example, this represents there's 98 PDB structures that match to that part of the sequence. And by clicking on the number 98, I can expand that number out into what's called a tree view. There can be multiple pages of this tree view. You can just step forward through them. And what we've done here is we've clustered them again now by what we call molecular conformation. So we can see, for example, that a large number of those structures consist of a single monomer of p53 by itself. And the ones that I'm showing currently in the 3D view there actually show a tetramer of p53 in complex with a DNA dimer. So by breaking these information down in this way, you're able to sort of easily get an overview of what's there and focus on the structures that are most relevant. So basically, Aquarium does two things: helps you find the structures that are out there, and then once you're there, it helps you learn from structures, in particular for people who don't generally use structures. There's a thing called the feature tab that consolidates information from Uniprot and Interpro on annotations of the feature. And the whole idea of Aquarium really was that I could do this, just click on a feature lane, these are annotations of the sequence, map those annotations onto a 3D structure, and have that as a coloring scheme. For this particular protein, there's a really interesting set of features that map the mutations that have been observed over 80,000 different cancers. And by clicking on this lane here, which contains the 5 residues in p53 that have had the largest number of distinct mutations across 80,000 cancers, I'm able to actually sort of map them, and we saw quite interestingly that they map into a very small space in 3D. Anyway, that's Aquarium, and that's the demo in 3 minutes. The idea of the resource is to make structures accessible to people, as I said, who don't generally use them, typically biologists. There's some wild and wacky structures out there. Actually, I came across one from the keynote talk. This is RAS, just happened to come across that. So the idea is help you explore the 3D database in a more natural way. And really what we want to do is we want to give people their own quick and once in a moment by making 50 million models accessible to the public. Most of them have never been seen by human eyes before. There's discoveries been made there. But to get onto the dark proteome, we spent a lot of time developing that resource and spent years looking at that view. And I realized some years ago, if there's a region where there's no structural match, it's a much stronger statement than just saying we don't know the structure there. Actually, we know that that region of sequence is not detectably similar to anything that we know the structure of. So it's really unknown in terms of structure. And so this was the basic insight that led to this dark proteome paper. And it's also an interesting observation that sometimes when you visualize stuff, it's not what you're expecting to see. It may not be necessarily the most important results, what comes out. So to build up the dark proteome, again, let's start with one prototypical protein into C-terminus. Aquarium maps trivially any region of any protein that is directly in the PDB. It also then matches any region of that protein that matches something in the PDB. Let's call that a gray region. And that leaves regions in between. We call them dark regions, where we don't know the structure. Now, if we look at eukaryotes, and we take all eukaryotic proteins end to end, 170,000 of them in SwissProt, and look at the number of amino acid residues that are actually covered directly by an entry in the PDB, it's about 4%. Okay? Remember, this is SwissProt, so we are overrepresented in the number of, or the percentage of coverage in the PDB. And based on that 4%, using Blitz, we're able to model something like 50% of the proteome in SwissProt in eukaryotes. It's quite impressive. Remember that there's a range here from things that are very similar to something in the PDB all the way through to some things that are very distinct, and so that's the reason for the gradient there. But at some point we have to draw the cutoff. It's about 50%. And this dark proteome region, about a third of that actually is not dark regions, but the dark region extends to cover the entire protein, and we call those dark proteins. If we compare these numbers for the other domains of life, we see overall eukaryotes are somewhat similar to viruses, although viruses have a larger fraction of dark proteins. And interestingly for Bacteroarchaea, we actually know somewhat less about their proteome, but we actually predict somewhat more. simple domains of life tend to reuse more commonly used architectures. So let's explore the dark proteome a little bit. So this is what we call a darkness profile. Again, this is for eukaryotes. So we've just taken the 170,000 sequences, measured a darkness score, the percentage of dark residues, and then counted the frequency of which a protein has that score. So we see by mode distribution, most things tend to have relatively low values, but there's actually a big peak of 100% darkness. Median value is 28% darkness, so eukaryotic proteins, about a quarter of the protein is dark. But just remember that and compare it to this graph, which is the graph of the disorder values across those same set. So actually I've got two different ones for the completely dark proteins, non-dark, but it doesn't really matter. The median disorder for eukaryotic proteins is somewhere between 6 to 10%, significantly less than the amount of darkness. So most of the darkness in proteins is not accounted for by disorder. Another way of seeing this, sort of turn those on the side, and this is a plot now where I've got every single protein, 170,000 of them, with the darkness and a disorder value. And basically what you can see is that along the diagonal here, most points lie above the diagonal, that is to say darkness is a larger value than disorder. So again, most of the disorder is not explained, most of the darkness is not explained by disorder. Another way of viewing this is what's called a linear diagram, sort of like a Venn diagram, flattened out. And there's another explanation that we could have for the dark proteome, which could be that some of those regions could be transmembrane, because we don't know the structure of many transmembrane proteins. This particular set is looking at all dark proteins, so only those things with 100% darkness, and only those things that have got high levels of more than 25% of residues disordered or transmembrane. And that seems to account for maybe half, but in eukaryotes at least, half of the dark proteins, we don't know why we don't know their structures, at least according to the disorder and transmembrane. Actually, that's a larger number if we compare the other domains of life. So a lot of the dark proteins and the dark proteome is unknown. What can we say about it? Well, if we focus on dark proteins, remember those are the things with 100% darkness. One thing we can easily measure is amino acid length, and we might expect they might be shorter. Indeed, there is they are significantly shorter in amino acid sequence, but not greatly, they're not ridiculously small. One very striking effect is, this is on the x-axis counting the number of similar sequences that we find in uniprot. So we see a big difference in the dark and non-dark proteins. Dark proteins have much fewer sequence relatives in uniprot. Probably not expected, probably expected considering how we calculated these data, but still a very strong observation. The next one was really unexpected, which is we went to the string database that aggregates together different evidence about protein-protein interaction, and we found that for non-dark proteins, eukaryotic proteins, the median number of interaction partners in string is 10 and 0 for dark proteins. So in other words, not only don't we know the structure of these things, but we don't know what they interact with. And I often use the analogy of, it's like drawing, if I was to draw a map of the rainfall level of a country, and I was to draw another map of the same country with the vegetation, those two maps would overlap a lot. They'd be quite strongly related. And similarly, we see with our knowledge of structure, or lack of knowledge in this case, correlates with our lack of knowledge of their interactions with other things. And similarly for many other functional properties. So for example, where a protein occurs in the cell. So many of the dark proteins, we really don't know anything about them. The unit point annotation is either weak or not existing. But for the subset where we do have annotation of subcell location, we find an overrepresentation of dark protein in the secreted, amongst secreted proteins. That's the strongest overrepresentation. Next is the endoplasmic reticulum, and the only location that was underrepresented in dark proteins was the cytoplasm. So we are on our way towards a complete knowledge, if you like, or more complete knowledge of the cytoplasmic proteins. So this is a little bit surprising that the secreted proteins had this strong overrepresentation. I'm not saying that all dark proteins are hanging out in one group somewhere and they're all secreted. Most dark proteins, well, the functions of dark proteins are doing lots of different things, but a subset of them are secreted. And again, in many cases, we really don't know where they are. Another way of looking at this is to do what we call a tree map. And here we've taken uniprot annotations for these dark and the non-dark proteins and compared them. Again, this is eukaryotes. And what I've done is draw a rectangle region for every functional term that's either over or underrepresented amongst dark proteins, and scaled the area by the minus log p, so the larger area is the more significantly over or underrepresented term. If we look at the tissue specificity of dark proteins, those are the ones in gray, associated with tissues associated with secretion, and 3 tissues where they're underrepresented. One is the red blood cell, which is primarily cytoplasm, so that relates to the previous observation, and the 2 other tissues which were too small to put text onto. One was the tissue called non-specific, and the other was a tissue called ubiquitous. So that's where dark proteins are underrepresented. Also overrepresented with domains that contain many cysteine knots and quite a few post-translation modifications, including many that relate to secreted proteins. So a consistent picture is emerging for at least a subset of these. We spent a lot of time on this paper. It was about, I think, 3.5 years thinking. I'm not saying we got everything right, but I think it makes an interesting read. Now we have another problem. I always say in biology we have 2 problems. First, we have to discover something new, which is hard enough, but then having discovered it, we have to tell a story. And what I found is visualization can help in both cases. So in my lab, I'm really excited about this new initiative in the last 2-3 years, where we're actually creating these molecular and cellular scale animations to tell stories about science. Great, but having put my money where my mouth is, and we've created a number of these animations, you can check them out on this website. Now we have the dark proteome, and we want to tell that story to a general audience as well. That's kind of tough because, again, how do we visualize the structures we don't know? Well, here's a first working draft, and I will get your help. This is a paper we have in progress on the human dark protein specifically, so that's sort of the numbers for human. And rather than show that as a pie graph to communicate to a broader audience, we probably need artistic ideas. And so actually we submitted to the Art and Science a poster here. One of our collaborators had a baby, and we sort of mapped onto them the approximate level of knowledge of the dark proteins. And now that you understand the background story behind this artwork, I'd encourage you to vote for it. So that's it. Just to convince you, we do other things, lots of other tools that are out there. Please check out the lab. But thanks for your attention. Happy to take questions.